So, this is my my drawings for my transcription, translation, modeling, deep dive assignment. Okay. Uh, so this is the double helix DNA strand, right? And then this is like a clear image of the double helix DNA strand, and this is my RNA strand. So basically, in our DNA strand, like we have the four letters. So T is thymine, C is cytosine, A is adenine, and G is guanine. And guanine pairs with cytosine, adenine pairs with thymine, and vice versa. Okay. So we're gonna talk about like the steps. So the first step is transcription, and there are like three main steps, which are initiation, elongation, and termination. A section of a DNA called the promoter instructs where and when transcription occurs. So let's say this, where it says transcription will occur, and then the MA polymers would unzip the two DNA strands. Then comes elongation, which is when an MA strand is created, and complements of the DNA strands, which would be called the template strand. So let's say this is a template strand, and uracil RNA strand that would complement it. The MA strand looks very identical to the non-template strand. However, one difference between these two strands is that thymine is replaced with another nitrogen base called uracil, which is right here. Um, so that's like one difference to keep in mind between like the DNA and the MA nitrogen bases. And then the last step is transcription, which is termination, which signals when transcription stops. So at this step, the newly created MA, which is currently attached to the template strand, would separate the DNA strand, and actually this MA strand goes through a couple moderations, removing sections that will not be able to be translated. And then the MA is transported out of the nucleus. So like the reason that is, it's because it performs translation in the ribosomes, and basically what that does is it builds codons, and then it makes proteins. So codons are the three nucleotides for that.